Namaskara Karnataka, welcome all of you to Vedantu Karnataka. Hegidira ellaru chennagidira. So we finished yesterday molecular basis of inheritance part 1. Ivat now part 2 madana. So last class alli ನಾವು search of genetic material DNA ಬಗ್ಗೆ RNA ಬಗ್ಗೆ general idea ಮತ್ತೆ structure ಬಗ್ಗೆ detail ಆಗಿ ಓದಿಸಿದ್ದೀನಿ. So class 12 students CBSE ಆಗಿರಲಿ PUC students, this is the best class that you can watch for molecular basis of inheritance. Everything I have broken down and taught you. So please watch this lecture and share it with your friends if they are having trouble with this chapter. Okay, please let me know in the chats if I'm audible and visible to all of you. Let me also just check. CBSE agarli PUC. Yes, very visible, very audible, very nice. Okay, come on. So start madana students class. So ivattu ಏನು ಮಾಡೋಣ, ಇವತ್ತು we are going to talk about replication, transcription, ಎರಡು main process na we will try to understand. Okay, so nanne class alli we have completed the structure. We have learned about what is nucleic acids and all of it, right? On the overview kottini illi. Okay, so usually nan class alli on 5 minutes revision irutte previous class do, so that it becomes easier for students who are joining in late also. They will be able to like, oh you ma'am finished so much, I'm leaving up. Many students what they do no, ಅವರು ಫಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಬರಲ್ಲ, ಸೆಕೆಂಡ್ ಕ್ಲಾಸಲ್ಲಿ ಬಂದ್ಬಿಟ್ಟು ಅಯ್ಯೋ ನನಗೇನು ಅರ್ಥ ಆಗ್ತಾ ಇಲ್ಲ, ನಾನು ಬಿಟ್ಬಿಟ್ಟು ಲೈಕ್ ಐ ಜಸ್ಟ್ ಲೀವ್ ಇಟ್, ಐ ವಾಚ್ ಇಟ್ ಲೇಟರ್ ಅಂತ ಆ ಚಾಪ್ಟರ್ ಫುಲ್ ಸ್ಕಿಪ್ ಮಾಡ್ತಾರೆ. ಸೊ ದಟ್ಸ್ ವೈ ವಿ ಗಿವ್ ಎನ್ ಓವರ್ವ್ಯೂ. ಓಕೆ, ಎಸ್, ಹಾಯ್ ಉದಯ್, ಹಾಯ್. ಸೊ ಸ್ಟೂಡೆಂಟ್ಸ್ ಹಿಯರ್ ಎಸ್ಟರ್ಡೇಸ್ ಓವರ್ವ್ಯೂ ವಿ ವಿಲ್ ರೈಟ್. ಸೊ ಈ ಬೋರ್ಡ್ ಅಷ್ಟು ಚೆನ್ನಾಗಿಲ್ಲ, ನಾನೇನು ಹೇಳಿದ್ದೀನಿ. ಸೊ ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ವಿ ವಿಲ್ ಗೆಟ್ ಅ ಬೆಟರ್ ಸ್ಟುಡಿಯೋ, ದಟ್ ಟೈಮ್ ವಿ ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ರೈಟ್ ಪ್ರಾಪರ್ಲಿ. ಓಕೆ, ಚಲೋ. Okay, overview. So overview is going to be whatever you studied yesterday. So those who know, okay, those who were there in the class, please try to answer so that it becomes easier for us to for me also to, okay, that students have understood. Okay, so yesterday we learned about nucleic acid. Okay, we learned about nucleic acid, and which are the two nucleic acid students? DNA ಮತ್ತು RNA. Okay, even if you are hesitant to type it in the chat box, just tell it out loudly in your home. Okay, if someone is listening, let them listen and feel happy. Okay, and if you're new to the channel, you know the drill. You have to subscribe to the channel and quickly smash the like button, because the only thing that we get, okay, as Guru Dakshana is your love through comments, through messages, through likes. Because namak ಬೇರೆ ಏನೂ ಇಲ್ಲ. Like offline class ಇದ್ರೆ I can see your faces and understand if you're all understanding. I have taught before offline also. So there I will look at, like, let's say I have taught classes where they had like 80-90 students also. I know all the 80-90 people's reaction I will be able to understand. But now I will not be able to understand. So how will I know that you are enjoying the class? By liking the session and giving me all the energy that you have. Okay, so nucleic acid students, DNA and RNA. So here major thing what you have to remember is that DNA, okay, in DNA the second carbon, tumba important idu, today's class also we will be talking about this. Second carbon has a hydrogen, and on RNA the same second carbon has an OH group. That is why this becomes deoxy. Okay, this becomes deoxy and this becomes just ribo. Okay, this becomes just ribo. Now talking about their structure, their structure is made up of, what is their structure made up of? Their structure is made up of nucleotide. Okay, nucleotide is made up of what and all components, students? Bega chat box alli, pentose sugar phosphate group. Okay, how did I ask you to remember? NPP, you remember. Okay, NPP and nitrogenous base. Okay, nitrogenous base. Understood? Pentose sugar means 5 carbon sugar. Okay, 5 carbon sugar. Clear? Phosphate group, normal phosphate group, matte nitrogenous base. Enide nitrogenous base alli purine matte pyrimidine. Okay, purine matte pyrimidine. I know most of you would have gone to college. So you will be thinking, "Ayo, why did this ma'am keep class at 2:30? We are all in college." Okay, no problem. You can watch this lecture later. Okay, recorded. You can watch this. Okay. Now look here. This is what we have studied. Now here we have understood that when we talk about purine, okay, we have to remember two people. Who are they? Adenine and guanine. Okay, adenine and guanine. These are the two people you have to remember. When you talk about pyrimidine, right? When you talk about pyrimidine, you have to remember about cut. Cytosine, uracil and thymine, right? Cytosine, uracil and thymine. Ivaga next most important things in students about the salient features is very, very important. Most important is Chargaff rule. Okay. Nanne on topic miss madidini students. Salient features ಬಗ್ಗೆ ಮಾತಾಡೋ ಟೈಮಲ್ಲಿ you have to remember about Watson, Crick, Wilkins and Rosalind Franklin. These 4 people were the one who understood that our our DNA looks like a double helical structure. Okay, so we have to understand that DNA looks like a double helical structure. How did they get to know? By doing X-ray diffraction. So by doing X-ray diffraction, they understood that yes, DNA is a double helical structure, and that double helical structure salient features is what you people study. So ad miss madidini nanne. So just remember that. Okay, Chargaff rule accordingly in students, ratio of purine should be equal to the ratio of pyrimidine. Alva? Okay, ratio of pyrimidine. Correct? So, illi we should understand that A will always bind with T. Okay, at Gopika Ma'am's class. So, Gopika Ma'am's class. Okay, G will always bind with C. Okay, this is the rule that we have studied. Now, other than that, what do we know? We know about nucleoside and nucleotide. Correct? Nucleotide. These two things we know. Nucleoside andre eno? Ma'am, simple ma'am, phosphate iralla ma'am, those other two will be there. So, we can write pentose sugar or we will just write sugar. Sugar plus NB is going to be the nucleoside. Okay, sugar plus NB plus phosphate is going to be nucleotide. Okay, nucleotide. Clear to all of you? Yes or no? Okay, this was one overview that we studied. Other than this, we also studied about the experiments as well. So, do you want me to write that as well? So, again, let's just, okay, 1 second. This overview will help you revise, and if you're a student who has missed the first class, it's going to come very handy for you, this notes. Okay, so students, yes. So, we learned about nucleoside and nucleotide. Then we studied about bonding. Bonding is the most important here also. Remember, 2 bonds you have studied. What is the first bond you studied? Sugar mattu nitrogenous base. Alva? Sugar mattu nitrogenous base between allon bond agute. A bonding ಏನು ಕರೀತೀರಾ? Glycosidic linkage ಅಂತ ಕರೀತೀರಾ. Okay? Glycosidic linkage. So, how did I ask you to remember? Sugar means glucose, so glycosidic you can remember, okay? Glycosidic linkage you can remember. Hello, I told you hello, Uday. Okay, quickly mark your attendance as well. Like Madi video na. Okay, glycosidic linkage. After that, what did we study, students? We understood that if this fellow wants to, like in the sense one more fellow, who is that? If a phosphate wants to attach, then what will be the bond form here? Phosphoester linkage. Okay, phosphoester linkage. Okay, so remember like this, phosphate, phosphoester. Okay, these are the 2 bonds that we have studied. It full on dot are considered madi, students. This full thing na on dot are considered madi. Okay, the whole thing. Okay, the whole thing is considered as a dot. Like this. Okay, so this is one nucleotide. Yes ma'am, it is one nucleotide. So how am I going to make the entire nucleic acid? I will need many nucleotides, alva. So one nucleotide, another nucleotide, another nucleotide, another nucleotide. Okay, like this many nucleotides will be there. Now if between them, if I want to make a bond, okay, if I want to make a bond between them, then that bond is going to be called as phosphodiester bond. Okay, phosphodiester bond. Tumma important. Diester. Diester andre 2 nucleotides madhyadal form agadu. Okay, form agutte 1 and nucleotide 2. Okay, nucleotide 2. So 1 and 2, between them the bond will be formed. Clear to all of you? Yes or no? Yes. Hi Daniel. So this was the overview that we learned yesterday. Then we learned about search for genetic material. Okay, search for GM. I'm writing genetic material. Don't tell search for Gopika Ma'am. Okay, search for genetic material. Students, we started with Griffith's experiment. Griffith's experiment tumba easy experiment. So in Griffith's experiment, he spoke about one thing that was called the transforming principle. Okay, transforming principle. Okay, here he took Streptococcus pneumoniae. Remember that 2 strains were there, 2 colonies were formed, 2 strains were there, one was virulent, one was non-virulent. Okay, and one was virulent because of the mucopolysaccharide coating. Okay, so transforming principle bage he spoke. Sorry, all the languages I mixed. Okay, next one was the biochemical nature. Okay, biochemical nature. So biochemical nature, what are they talking about? They are going to talk about what? Okay, yes, uta I had on the way, in the auto only I had lunch. Okay, so biochemical nature, what happens, students? Here he's tried to find, is RNA the transforming principle, is it DNA or protein? Okay, or protein. So they wanted to know which among this was the genetic material, and they concluded that DNA is the genetic material, but it was not accepted by anyone else. Okay, they concluded that DNA is the genetic material, but no one was happy with that. Then came Hershey and Chase experiment. Okay, Hershey Chase experimentally main hero yaro bacteriophage, virus that infects bacteria. Okay, and the bacteria we took was E. coli, bacteriophage. Half of the words barala students, boardally. Okay, bacteriophage. So they proved that DNA is the genetic material. Okay, done. So the overview is done. Ivat class start madana. So molecular basis of inheritance part 2 matta iddivi. Students, this chapter is very important for NEET, KCET, for PUC. Every level this chapter is the most important chapter. Okay, so next, so I told you last time also that some of you were asking, ma'am, do we have a Vedantu learning center? Yes, we have an offline center in Karnataka, Bangalore. It is in JP Nagar. Okay, it is in JP Nagar. So if you are a person who is in Bangalore and who can travel to JP Nagar or is in and around JP Nagar, then you can go there, book a visit, and you will be able to know what are the facilities that are provided, how are the classes being taken. So you will get an overview of what exactly happens in Vedantu Learning Center. Okay, na? So yes. So if you are nearby, you can definitely run and go. Ivaga first topic start madana students, RNA mattu DNA bage. Okay, RNA and DNA, all of you. RNA and DNA. So yesterday's class I already told you that RNA is not a genetic material for all the organisms. It is a genetic material only for viruses. Now DNA on the other side is the genetic material for most of the organisms. Why did this happen? One reason irbekalva. So reason enandre students, very simple nodi. So when I told you that RNA has in the second carbon a hydrogen, RNA has OH, okay, OH in the second carbon. I was not just telling about the structure, but more than that, this OH which is there in the RNA is highly reactive. It's highly reactive. Students, because oxygen normally highly reactive agirutte, so idu highly reactive. Okay na? So here what is going to happen? Understand, on DNA on the other side, okay, we have DNA whose second carbon has only a hydrogen. Because of which this is least reactive. So do you want your genetic material to be highly reactive? No ma'am, I do not want my genetic material to be highly reactive. I want my genetic material to be least reactive. Alva. Second reason in students, very simple. So thymine is present, right? Thymine is present in DNA. Same thing, thymine is replaced by uracil in RNA. Alva, yesterday I taught you this. So here this also makes the stability of DNA more. Thymine makes the DNA more stable compared to RNA who has uracil. So these two reasons are there. Okay, that means your DNA compared to your RNA, is more stable. You obviously want a genetic material who is going to be stable whatever happens outside. Okay, whatever is happening outside, you have to be more stable inside, right? More stable, less reactive, and most importantly, students, DNA does slow mutation. Okay, slow mutation. You do not want to sleep as a human and wake up as a penguin, we tell a joke. Okay, so like that only, we want slow mutation. We do not want fast mutations like RNA. Okay, so these three reasons made DNA the genetic material for most of the organisms. Okay, for most of the organism, DNA was the genetic material because of these three reasons. Okay, ivaga NCERT on point quoted, like few points have been given, so let's just check that as well. See, a molecule that can act as a genetic material first should fulfill the following criteria. Very simple. Now, if you want to stand for election, okay, you have certain criteria to go through. If you want to vote, okay, you should be 18 years and you should be a citizen of India. Okay, all that criteria are there. Why? So our criteria follow madbittu one set of people only will be able to vote. Illandre everyone will be trying to vote alva. So adikke one proper criteria idre, there will be set of people who will stand for the politician role or MP role or MLA role. There will be certain people who are going to look for jobs. So ella job allu ella ಒಂದು medium allu ಒಂದು what is that, boundaries are there, criteria are there. ಆ ತರನೇ if DNA or RNA wants to become the genetic material, ಅವ್ರಿಗೂ one set of criteria are given. Okay, yes. So ಇಲ್ಲಿ ನೋಡಿ, it should be able to generate its replica, andre it should be able to undergo replication. So ಇಲ್ಲಿ ಯಾರು points ಕೊಡೋದು students? So ಇಲ್ಲಿ ನೋಡೋಣ, ಇನ್ನೊಂದು ಚಿಕ್ಕ box we can draw. Okay, ಒಂದು ಚಿಕ್ಕ box we can draw. DNA ಮತ್ತು RNA. Okay, so one point, first point we'll give it for both of them because both of them can do replication. Okay, 1 point for both of them. Okay, 1 point for both of them. It should be stable chemically and structurally. Is it stable chemically and structurally? No ma'am, RNA is not chemically or structurally stable. So I give 1 point to DNA and no points to RNA. Okay, I give 1 point to DNA and no points to RNA. It should provide the scope for slow changes, that is mutation, that are required for evolution. Ma'am, here same thing is going to happen. DNA is going to get 1 point, RNA is going to get no point because RNA does faster mutation. Okay, it should be able to express itself in the form of Mendelian characters. So this both of them are going to do. So I'll give 1 point for both of them. Okay, 1 and 1. So ill nodi students, let's just do the maths. Okay, let's just do the maths. This is going to be 2 points and this is going to be 4 points. So who should be the genetic material, DNA or RNA? Ma'am, it is understood that DNA should be the genetic material. Okay, DNA should be the genetic material. So 4, obviously, because why? It can form its own replica. It's stable chemically and structurally because of 2 reasons. Because of the absence of the oxygen in the second carbon and the presence of thymine instead of uracil makes the structure more chemically and structurally stable. Okay, it should provide a scope for slow changes. DNA goes for a slow changes, it goes through slow mutation, but on the other side RNA goes for faster mutation. Okay, it should be able to express itself in the form of Mendelian characters. Artha? Understood all of you? Clear or not? So these are the criteria that is given for to be the genetic material. Okay, so they have given more. Let's just read and understand the paragraph of NCERT. So ee class admele nimage NCERT odak separate time you do not have to find. Idalle I am making you read NCERT as well. So make sure that you sit and watch the entire class. Okay, now the genetic material should be stable enough not to change. Okay, stable enough not to change with different stages of life cycle. Alva, when I am a child, I do not want to be like some other organism, then I become adult, some other organism. When I reach my old age, some other organism. We do not want our genetic material to do that kind of drastic evolution. Okay, changes. We want slow changes. Okay, so you'll notice students, life cycle, age, or with change in physiology of the organisms. Stability is one of the properties of genetic material that is very evident. Stability is one of the properties of genetic material that was very evident in Griffith's transformation principle. That is, ashtu heat madamele, okay, heat killed estrate. After heating it to an extreme temperature, still DNA was not denatured, DNA was not harmed. DNA did transformation. That means any organism which is around will pick up the genetic material, that is called as transformation, which is mainly seen in bacteria. Bacteria, the least sexual reproduction time when we teach you transduction, conjugation and transformation. Adal one step is transformation. And if in case any genetic material is lying around nearby and due to any reason the cell has got damaged, our DNA or pick up ಮಾಡ್ತಾರೆ. ಅದೇ ಆಗಿರೋದು transformation ಅಲ್ಲಿ. So Griffith's transforming principle, okay, itself that the heat which killed the bacteria at least did not destroy, okay, at least did not destroy some properties of the genetic material. See students, some properties obviously would have got damaged because of the extreme heat, but the properties that is required for a genetic material to be, be able to behave, that kind of characters were still there, okay. This now can easily be explained in the light of the DNA, that the two strands being complementary if separated by heating come together when the appropriate conditions are provided. Example nodi, like Chintu Mintu idare, okay. Chintu Mintu ge they really like to, they are like best friends, okay. They like to go for movie together, they like to bunk classes. ಆ ಥರ ಒಂದು ಕಾಂಬಿನೇಷನ್ ಇದ್ರೆ ಟೀಚರ್ಸ್ ಆಬ್ವಿಯಸ್ಲಿ ಅವ್ರನ್ನ ಕ್ಲಾಸಲ್ಲಿ ಸಪ್ರೇಟ್ ಮಾಡ್ತಾರೆ ಅಲ್ವಾ. ಅದರ್ವೈಸ್ ದೇ ವಿಲ್ ಬಿ ಲೈಕ್ ಯು ಪೀಪಲ್ ಆರ್ ಡಿಸ್ಟರ್ಬಿಂಗ್ ದ ಅದರ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಸೊ ವಿ ಹ್ಯಾವ್ ಟು ಸಪ್ರೇಟ್ ಯು. ಅವ್ರು ಏನು ಮಾಡ್ತಾರೆ, ಟೀಚರ್ class in, like let's say that period got over, the moment the teacher leaves, Chintu Mintu will come back together and they will start sitting together. Adhe tarane DNA, if they are complementary to each other, and one strand has adenine and another strand has thymine, guanine, cytosine, obviously they will have a tendency to come back to each other, right? So same way, if I heat it extremely, the strands are going to separate, but when the condition becomes normal, they will come back together anta explain madidare nodi. Further, the 2OH group, okay, the 2OH group present at every nucleotide in RNA is a reactive group and makes RNA less stable and easily degradable. RNA is also now known to be a catalyst. Catalytic kendra, it increases the rate of reaction, increases the rate of reaction. Okay, increases the rate of reaction but does not directly take part in the reaction, okay, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. Clear to all of you why most of the organisms have DNA as the genetic material, okay. Now, in fact, one more, I told you, students are direct agi hinge kelbodu, what is the reason that DNA is the most stable? So ನೀವು ಸುಮ್ಮನೆ replication ಮಾಡ್ತಾರೆ, slow mutation ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ the points are different. They are talking to you about why DNA is stable or why DNA is considered as the most stable genetic material. ನಿಮ್ಮ explanation ಬಂದ್ಬಿಟ್ಟು first about the OH and the absence of oxygen, second ಬಂದ್ಬಿಟ್ಟು you have to write about the thymine. ಅದಾಮೇಲೆ additional points ಗೆ ನಿಮಗೆ whatever else you can write, okay. So question matte answer, keywords you have to look and you have to answer, okay. Now, in fact, the presence of thymine at the place, okay, presence of thymine at the place, one second, yeah, the presence of thymine at the place of uracil also confers additional stability to DNA. Detailed discussion about this requires understanding the process of repair in DNA, and you will study this in higher classes, okay. So additional stability is given by whom? Obviously the presence of thymine, okay, presence of thymine. Now, both DNA and RNA are able to mutate. I told you already that they both can mutate, no doubt in that. In fact, RNA being unstable mutate at a faster rate. So I do not want a genetic material that will mutate at a faster rate. I want a genetic material who will mutate at a slower rate and do not change the evolution completely, okay. So consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of protein, hence can easily express the characters. DNA, however, is dependent on RNA for the synthesis of protein. So, we cannot tell that RNA is completely useless. Because RNA is helping in forming the protein. Alva, now, nanne central dogma when we were studying, namagartha aitu that RNA can convert to protein with the help of a process called as translation. So, RNA banbitu messenger, it can behave like a messenger, adapter, catalyst. So, bere ella processigu RNA is helping DNA, but adike genetic material agak properties ಸ್ವಲ್ಪ ಕಡಿಮೆ. Okay, criteria ಅಲ್ಲಿ ಸ್ವಲ್ಪ they are little different. Okay, so RNA can directly code for synthesis of protein, hence can easily express the characters. DNA, however, is dependent on RNA for the synthesis. The protein synthesis machinery has evolved around RNA. Clear to all of you? Yes or no? Give me a thumbs up if all of you understood. Give me a thumbs up. Yes. Give me a thumbs up if all of you understood quickly in the chats. Colors change madana. I'll just change the colors by then so that it becomes easier. What happens, students? It's not changing. Yes. Okay. So look here, DNA replication. Replication means the ability to form their own copies. Okay, own replica. Okay, own replica. That is called as replication. Correct. Now, how do they form that, ma'am? Wait, I will come into that. Okay. One second. Okay, own replica. Now, how do they form that? I will show you. So, students, here our DNA replicates in a semi-conservative manner anta explain ಮಾಡ್ತಾ ಇದ್ದಾರೆ. Okay, ಹೆಂಗ್ ma'am DNA replicates in a semi-conservative manner. Okay, semi-conservative, correct. Now, what is semi-conservative? Semi-conservative explain ಮಾಡಕ್ಕೆ first ನಾವು conservative ಏನಂತ ನೋಡಬೇಕು. Okay, so I am going to explain all of this with the help of a diagram. So, let's assume this is our template strand. Okay, we can also call this as a parent strand. Okay, this is our template DNA strand. Okay, you got one DNA strand. I call this the template or I call it the parent strand. Okay, parent strand. Now, I am going to use this parent or template to form new copies. Okay, to form new copies. So, nan first ಏನು ಮಾಡ್ತೀನಿ ಇದನ್ನ separate ಮಾಡ್ತೀನಿ. For that we have to utilize different enzymes which you will be studying later. Okay. Okay, now I separated these two strands. Yes, ma'am. So, I label it, let's say 5' to 3' and 3' to 5'. Both the strands are separated. They are anti-parallel polarity. Now, I am going to use this template to form my daughter strand. So, they are obviously going to be complementary. Yes, ma'am, they are going to be complementary. I got my two strands, correct? So, I am going to label this. Obviously, this is 5' means this will be 3', this will be 5'. Okay, this will be 5', this will be 3'. Alva? Correct agidya? Yes, ma'am, correct agide. And these people are going to be my daughter strands or my newly synthesized strands because I am forming my own replica. So, obviously, I need these strands. Okay, ille en agutte students. Normally, conservative technically, okay, conservative technically ಏನು ಆಗುತ್ತೆ ma'am? Simple, ma'am. The parents are going to go back together, alva? Parents will go back because they are used as a template, alva? Parents will go back and obviously the daughters also will go back. Okay, this is conservative. This is conservative. Okay, this is the entire conservative technique. So, ಏನಾಯ್ತು students, ಇದನ್ನ template ಆಗಿ ಯೂಸ್ ಮಾಡಿದ್ದೀನಿ. Separate ಮಾಡಿಬಿಟ್ಟು I form the daughter strands and then this is what is going to happen. Okay, this is what is going to happen. Now, what is happening in our DNA is a little different. Okay, it is little different. Idana simple example you can remember. See, imagine this is your Xerox copy. Okay, this is ma'am's Xerox copy. Okay, ma'am's copy. Let's say I'm giving you the original copy of my notes. You will go and take a Xerox. Okay, let's label this as ma'am's copy. Okay, because some students find it very difficult to understand this, but it's very easy. See, let's consider this is the ma'am's copy or the original copy. Okay, this is the original copy. If I give you this original copy, what will you do? You will go to a Xerox shop and you will take a copy of it. So, this is going to be your Xerox. Okay, yes ma'am, this is going to be my Xerox. Let's say this is one sheet, you are taking a Xerox of this. This is one sheet, you are taking a Xerox of it. After some time, what you will do? You will pin all ma'am's copy. Okay, you will pin all ma'am's copy and just give it back to ma'am. On the other side, you will take your Xerox and pin it and go. This is the normal technique, conservative. Okay, but num DNA ಏನಾಗುತ್ತೆ students? Num DNA ಸ್ವಲ್ಪ different ಆಗುತ್ತೆ. I'll tell you why. Okay, I'll tell you why. Let's take the same example. Okay, let's take the same example. Just that we will remove the daughter strands. We will just change the color. Okay, yeah. Same thing is continuing. Parents, okay, parents, obviously they're going to act like templates and they're going to give me the new daughter strands. Okay, again, I'm going to get the new daughter strands. Yella process same continue agutte. Okay, same daughter strand. It'll 5 dash andre, it'll 3 dash barutte, it'll 3 dash andre, it'll 5 dash barutte. Okay, same thing here, it'll 5 dash, it'll 3 dash. Okay, na, done. ಇವಾಗ ಏನಾಗುತ್ತೆ ನಾರ್ಮಲಿ ನೀವೇನು ಮಾಡ್ತೀರಾ you will give back the copy, ma'am's copy and your Xerox separately. But you found a brilliant idea. What was the idea? You took one copy of ma'am, okay, one copy of ma'am's original and you took one copy of your Xerox and you mixed it. Okay, understood? Clear to all of you? Yes or no? Okay, yes. Aishu, I checked it. Aishu, it is a glitch. Okay, it is a glitch. Okay, don't worry. All that will be fixed. It is actually a glitch where, you know, some letters and names cannot be typed. Okay, that will be fixed. It is actually like that. Many glitches keep happening on YouTube. Okay, sometimes we can't type the numbers. Sometimes we can't type some letters. Okay, yeah. Come on. Okay, in Lodi students. So, ಈ ತರ ಮಾಡಿದ್ದೀರಾ ಏನು ಮಾಡಿದ್ದೀರಾ one ma'am copy ಮತ್ತು one parent copy you have mixed, sorry, one your copy and one ma'am's copy you have mixed, and this is called as semi-conservative. Understood? This is called as semi-conservative. Clear to all of you? So, let's write the same numbers here. Let's say this is from 5 dash to 3 dash. This is going to be 3 dash to 5 dash. Okay? Same thing here, 5 dash. Okay, this strand will be 3 dash. Okay, first 3 dash to 5 dash, 5 dash to 3 dash. Got it? This is how you're going to form. Okay, this is how you're going to get. That means this is a semi-conservative. Alva nodi students, semi-conservative. One is the parent strand, one is the daughter strand. Okay, or one is the template and one is the newly formed strand. Okay, so we call this semi-conservative and we call these strands as hybrid strands. Hybrid classes andre eno online nadita irutte, offline nadita irutte alva. Hybrid strands anta karitivi. Okay, hybrid strands anta karitivi. So this is how your DNA replication is going to happen. So with the help of colors, it is more easier to understand. Okay, it's more easier to understand. So this is conservative. Well, let's say it's ma'am's copy and the Xerox is separate. Here what is happening? One ma'am copy, one Xerox, one ma'am copy, one Xerox is happening. Okay, clear to all of you? Now let's go through NCERT. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication. So who came up with replication, this, what is the finding? It was Watson and Crick. They will ask you question. Okay, now to quote their original statement, that is as follows: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism. So, replication is otherwise, what is it? Copying mechanism for genetic material. Okay, Watson and Crick has quoted this statement andre avaru helidare in 1953 that when we looked at the complementary base pairing, we noticed that there should be one copying mechanism as well. Okay, now the scheme suggested that the two strands would separate, act as a template. Okay, act as a template for the synthesis of the new complementary strands. Okay, so our parent strands ಎರಡು separate ಆಗಿ they will act as a template. ಇದೊಂದು strand form ಆಗುತ್ತೆ, ಇದೊಂದು strand form ಆಗುತ್ತೆ. ಈ ಎರಡು strands separate ಆಗಿ ಹೋಗ್ಬೋದು, ಇಲ್ಲಾಂದ್ರೆ if they join together they are going to form the semi-conservative pattern which is present in our DNA. Okay, after the completion of the replication, each DNA molecule would have one, see listen carefully, after each of it what will they have? One parental and one newly synthesized strand. Clear? One parental, one newly synthesized strand. This scheme was termed as semi-conservative. Okay, semi-conservative. Clear to all of you? I hope this is super clear. Okay, I hope this is super clear. So this is what they are telling. One parent, one newly formed strand. Okay, one parent, or let's say in other words template. Okay, and one daughter strand. Okay, one daughter strand, or in other words newly synthesized strand. Okay, newly synthesized strand. Clear? Done, done, done. Okay, now students, Messelson and Stahl, okay, two scientists came and they were like, wait, let's try proving this with the help of an experiment. Okay, yes, we will try proving this with the help of an experiment. I want to see all of your attendance first. 1 minute. Let me see how many of you marked your attendance. Okay, all lazy, busy, conjuice people. Students, yak students attendance marke madilla? Huh? Nim friends ಹೇಳ್ತಾ ಇದ್ದಾರೆ, you are not listening to your friends only. Yes or no? Done, pakka? Okay, timing ಇವತ್ತು ಸ್ವಲ್ಪ I had to take early because ನನಗೆ NEET English ಅಲ್ಲೂ ಕ್ಲಾಸ್ ಇದೆ, ಸೊ ಅದಕ್ಕೆ ಸ್ವಲ್ಪ I had to take the class a little early. Okay, yes. Okay, cool. Got it? Come on. You people are not asking me if I had lunch. Only Uday asked me if I had lunch. Huh? Always asking each other, did you have lunch? Did you have lunch? Okay? Never ask the teacher anything. Never ever ask the teacher anything. Ma'am, you eat lunch, you don't eat lunch. What for us, ma'am? You come and take class. Is that the attitude? Huh? Okay, come on. Chalo, Messelson and Stahl experiment. I want all of you to listen to me carefully. If you don't listen to me carefully, you're not going to understand this. Okay? So students, we'll take one Petri plate. Okay? This is one Petri plate. Okay? And this is one more Petri plate. Okay? So first Petri plate. Here solutions etkonta idini. One is NH4Cl. Okay? Ammonium chloride. But illi heavy. Okay? This is a heavy isotope. Okay? Remember, it's not radioactive. It's just a heavy isotope. I hope all of you know what is an isotope. Don't ask me. Okay? It's a heavy isotope. Correct? Yes, ma'am. Second solution I'm going to take is going to be NH4Cl. Okay? But 14. I'm going to take 14. That is obviously not the heavier isotope. But again, this is the isotope, light isotope. Okay? Light isotope, but they're not radioactively labeled. Remember that. Okay? They're not radioactively labeled. 1 second. Ayyo. Light isotope. Okay, light isotope. Got it? Okay. Now I am going to take E. coli. Whom am I going to take? I am going to take a E. coli. Now, if I take this E. coli, what is the use, ma'am? Why are you taking E. coli? First of all, E. coli is a bacteria. Second of all, students, they can do replication. Tumba important ido. They can do replication in 20 minutes. 20 minutes ಅಲ್ಲಿ replication ಆಗುತ್ತೆ. So obviously ನನ್ನ experiment ಅಲ್ಲಿ ನನಗೆ full day lab ಅಲ್ಲಿ ಕುತ್ಕೊಳ್ಳೋಕ್ಕಾಗಲ್ಲ. So obviously ನನಗೆ 20 minutes ಅಲ್ಲಿ result ಬೇಕೇ ಬೇಕು. Okay? ಬೇಕೇ ಬೇಕು. Yes, Anirudh, I have taken your name. I hope you are happy. Okay? Naming ceremony is happening, students. Okay, now what is going to happen? I am taken E. coli. First, I will let the E. coli, okay, be here in your NH4Cl15, okay, heavy isotope. Okay, heavy isotope. So obviously students, ನನಗೆ replication ಆದ್ಮೇಲೆ ಏನು strands, what strands will I get from the first generation? So let's consider this. This is the first generation, no? First gen I am writing, okay. So in first gen, what am I going to get? Ma'am, it is understood that in the first gen, you are going to get 2 strands. Okay, you are going to get 2 strands which are N15 and N15. That means they are going to be having the heavy isotope, correct? First generation, I am going to get complete N15, N15. Okay, done? Yes. Now, I transfer the E. coli. Let's say I take the E. coli and I add it here. Okay, to your N14 light isotope. Light isotope. That this is going to be my second generation. That means after 20 minutes, I am seeing everything. Okay, 20 minutes. Obviously, I am going to get what? I am going to get a mixture. Okay, I am going to get a hybrid. Why am I going to get a hybrid? It is understood because my DNA is semi-conservative. So, I am not going to get an entire light strand. I am going to get a mix of light and heavy. Right, I am going to get a light and heavy after 20 minutes. Okay, so here what am I going to get in the next generation? Okay, next generation I am going to get one light and one heavy and one light. Correct? I am going to get a hybrid. Color ashtu differentiate ಮಾಡಕ್ಕೆ ಆಗ್ತಾ ಇಲ್ಲಲ್ಲ. Just give me a minute. Orange use ಮಾಡಿದ್ರೆ students, let me see. Orange is better, no? Okay, wait. I'll do it in orange, okay? Okay. So, light is going to be in orange. This is what I am getting after second generation. I am going to get half light, half. Okay. So, here what am I going to get? I am going to get N15 and I am going to get N14. That means I got hybrid. Okay, I got hybrid. How many strands will I get? 2 strands I will get, okay, because these both are going to act as a template, okay. So, I am going to obviously get 2 strands like this. So, I am going to get 50% hybrid, correct. So, this is what is happening, okay. Idella admele, they will be put into cesium chloride and they will be doing centrifugation of it. So, that also you have to remember here other than these people, okay. Cesium chloride for density gradient, okay, because here it is density, one is heavy, one is light, and density basis al ಮಾಡ್ತಾ ಇದೀವಿ. But Hershey and Chase experimentally it was radioactivity, so confuse ಮಾಡಬೇಡಿ, okay. This is based on density gradient, okay, density gradient, correct, density gradient. So we are going to take cesium chloride and we are going to do centrifugation. Centrifugation. Okay, centrifugation. Clear? Yes or no? Done, done, done? Okay, done. Done, 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 done. Now, I will tell you exactly what is going to happen at each point, okay? At each point, what is going to happen, I will tell you. I am erasing all of this. Students, this and yesterday's notes together, I will share it with all of you, okay? Actually, yesterday notification came, so I forgot to share this. Yes. So, what happened? We will see. Okay, what happened? We will see. Okay. Look here, all of you. Very simple. Okay. First, I told you, heavy isotope you are going to take. It is going to be blue in color. Okay. Colors, please remember. This is your NH15. Okay. N15, N15. Correct? This is what I got in the first generation. Obviously, these people are going to be heavy and we can consider them like the template, okay. Now, after 20 minutes, okay, I moved the E. coli to N14, okay, 20 minutes, what am I going to get? I am going to get, students, ee ಎರಡು strands separate ಆಗುತ್ತೆ. They are going to act as the template, okay, ಎರಡು strands separate ಆಗಿಬಿಟ್ಟು ಅದರಲ್ಲಿ ಏನು ಆ್ಯಡ್ ಆಗುತ್ತೆ? Obviously, the light, okay, DNA is semi-conservative, so they are going to form this, right. So, here what have I got? I am going to get 100% hybrid. Okay, 100% hybrid. Yes, ma'am, you're going to get 100% hybrid. Again, let's say after 120 minutes. Okay, after 20 minutes, let's say I separated these strands. 1, 2, okay, orange, orange. Okay, what am I going to get? Everywhere light is going to get added because again, remember, they are going to stay in the NH14 medium for longer. Okay, they are going to stay in the NH14 medium for longer. So, obviously, we are going to get more light strands. So, here what are you going to get, students? Here you will get 50% hybrid, 50% hybrid, and you are going to get 50% light. Okay, because now newly synthesized strands jasti irbeku. Parent strand alla jasti iradu. Obviously, newly synthesized strands is going to be, more. Okay. Ma'am, this is my favorite class. You're my favorite teacher. Thank you so much, Uday. Okay, so 50% hybrid students, 50% light. Artha aita? Okay. ಇದು ನೆಕ್ಸ್ಟ್ ಸ್ಟೆಪ್ಗೆ ಹೋಗ್ಬೇಕಂದ್ರೆ ಅಂದರೆ 20 ಮಿನಿಟ್ಸ್ ಆದಮೇಲೆ ಏನಾಗುತ್ತೆ ಮ್ಯಾಮ್ ಅಂದರೆ ಸಿಂಪಲ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಏನು ಮಾಡಬೇಕಂದ್ರೆ ಒಂದು ಈಸಿ ಟ್ರಿಕ್ ಹೇಳ್ಕೊಡ್ತೀನಿ. ಇನ್ನೂ ಒಂದು 20 ಮಿನಿಟ್ಸ್ ಆದಮೇಲೆ ಏನಾಗುತ್ತೆ, ಇಲ್ಲಾಂದರೆ ಒಂದು 40 ಮಿನಿಟ್ಸ್ ಆದಮೇಲೆ ಏನಾಗುತ್ತೆ, ಈ ಸ್ಟ್ರಾಂಡ್ಸ್ ಎಲ್ಲ ಸಪ್ರೇಟ್ ಮಾಡಿಬಿಡಿ. ಓಕೆ, ಎಷ್ಟು ಬ್ಲೂ ಸ್ಟ್ರಾಂಡ್ಸ್ ಇದೆ? 2 ಬ್ಲೂ ಸ್ಟ್ರಾಂಡ್ಸ್ ಇದೆ ಅಲ್ವಾ? ಎಷ್ಟು ಆರೆಂಜ್ ಸ್ಟ್ರಾಂಡ್ಸ್ ಇದೆ ನೋಡಿ? 1 2 3 4 5 6. ಓಕೆ, 1 2 3 4 5 6. ಎಲ್ಲ 6 ಸ್ಟ್ರಾಂಡ್ಗೂ ಲೈಟ್ ಸ್ಟ್ರಾಂಡ್ ಆ್ಯಡ್ ಮಾಡಿ, ಅಂದರೆ ನ್ಯೂಲಿ ಸಿಂಥಸೈಸ್ ಸ್ಟ್ರಾಂಡ್. ಓಕೆ, ಎಲ್ಲದಕ್ಕೂ ನ್ಯೂಲಿ ಸಿಂಥಸೈಸ್ ಸ್ಟ್ರಾಂಡ್ಸ್ ಇಫ್ ಯು ಆ್ಯಡ್, ವಾಟ್ ವಿಲ್ ಯು ಗೆಟ್? ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಗೆಟ್ ಹಿಯರ್ ನೋಡಿ ಇಲ್ಲಿ 25% ಹೈಬ್ರಿಡ್, 25% ಹೈಬ್ರಿಡ್. ಹೈಬ್ರಿಡ್ ಕಮ್ಮಿ ಆಗಿ ಬರುತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್. ಓಕೆ, ನ್ಯೂಲಿ ಸಿಂಥಸೈಸ್ ಸ್ಟ್ರಾಂಡ್ ಜಾಸ್ತಿ ಆಗಿ ಬರುತ್ತೆ. ರೆಸ್ಟ್ ಫುಲ್ ನ್ಯೂಲಿ ಸಿಂಥಸೈಸ್ ಲೈಟ್ ಸ್ಟ್ರಾಂಡ್ಸ್ ಅಲ್ವಾ? ಸೊ ರೆಸ್ಟ್ 75% ಲೈಟ್. Like that, if you take it to the next step also, you will get 87.5% light, 12.5% newly hybrid. Okay, so like this you can do the entire experiment. Now I'll show you what Messelson and Stahl did. Okay, so see, now come here. They grew E. coli in a medium containing N15, okay, N15 NH4Cl. N15 is a heavy isotope of nitrogen. As the only nitrogen source for many generations, the result was N15 was incorporated into the newly synthesized DNA because avrige bere eno option idilla, only N15 ittu. Okay, yes, okay ma'am, nim class ne nanage tumba ishta agta ide. Okay, thank you. Okay, yeah. So N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride. Why? Because obviously this was heavy isotope. So if I do centrifugation, I'll get the heavier particle on the pillet and supernate and I will get the rest. Nane, I have already taught you people this. Okay. So density gradient. Please note that N15, this is tumba important students. Okay. My God, my Konglish is becoming max pro. Okay. This is tumba important students. Okay. And it can be separated from N14 only based on densities. If I want to separate light and heavy, let's say I want to separate any substance, light and heavy substance, I will obviously do what? I'll wait for it to settle down. Ivaga I will have soil and water. If I want to purify the water, I will wait for the water to go up and the soil to settle down, right? Same thing here also. If I want to separate heavy and light, I will definitely do a centrifugation with the help of a density gradient, use cesium chloride. So I will get pillet and supernate. Okay, now then they transfer the cells into a medium with normal NH4Cl, okay, normal NH4Cl, and took samples at various definite intervals. As the cells multiplied, extracted the DNA that remained as double stranded helix. The various samples are separated independently on cesium chloride to measure the densities of the DNA. Okay, now students, experiment nodi, experiment munche I think some more explanation is there. Thus the DNA that was extracted from the culture one generation after the transfer from N15 to N14, that is after 20 minutes, because E. coli divides in 20 minutes, had a hybrid or a intermediate density. I have already showed you that. First generation in the now shift mada time ali we got hybrid because one strand banbit N15 itu, innon strand banbit N14 itu. So obviously understood that they are intermediate or hybrid, just like how we expected. Okay, and the DNA is going to be semi-conservative anta. Okay, now DNA extracted from the culture after another generation, that is after 40 minutes they have taken. Okay, that is second generation, was composed to have equal amounts of hybrid DNA and light DNA. Nanu helilva 50% hybrid matte 50% light anta. Okay, easy to understand. Now If E. coli was allowed to grow for 80 minutes, then what would be the proportion of light and hybrid density DNA molecules? Every similar experiment involving use of radioactive thymidine to detect, okay, idu separate to bere experiment. So, idu artha aitala? Okay, yes. Okay. Now look here, this was the Messelson and Stahl experiment, students. First our rain madidare N15 medium they have taken, that is heavy. Obviously it was not there for a longer generation, so both the DNA strands were N15. After 20 minutes, when they transferred it from N15 to N14, they got hybrid, which was complete, okay, complete 100% hybrid, okay, 100% hybrid. So this is what they got in the first generation. Adamele 40 minutes admele, okay, 40 minutes admele énayitu students, generation 2 alli they looked at the same amount of light strands and the same amount of hybrid strands they got. That means your answer will be 50% hybrid, okay, 50% hybrid and 50% light, correct, 50% light, alva. So this is what they got in the second generation, okay, this is the second generation. This is the second generation. Clear to all of you? Now again after 40 minutes if you try also, I told you, adana separate madbit maadi anta, okay. Now, is this clear? So students, avaru question keltare exam ige. Remember that they will ask you questions, okay, they will ask you questions based on the time. That, okay, if the E. coli is like, let's say after, you know, after 100 minutes, after 100 minutes what will you get, or after like 80 minutes what will you get. So for that you have to know the experiment properly. Remember that if they are asking for 80 minutes, that means each division takes around 20 minutes, no? So you have to get at least 4 generations. Okay, 4 generations you have to get. So like this you have to do the calculation. Clear? Yes? Okay, I will check the request here. Okay, clear to all of you? So E. coli after 100 minutes, 80 minutes. So simple ag madbekandre colors use maadi madbodu. Illa andre if you are like, no ma'am, I know the technicality of it, then you can continue doing it. Illa andre nan already explain madidini. Colors use madkond easy ag madbodu like this. Okay, like this, okay. Now if you separate the strands, you will be able to know how many strands are there. So if I separated, how many strands am I supposed to get in the second generation? 2 blue I should get, 2 red I should get, right? 2 blue, 2 red. Elladukku light strand if you add, this is a newly synthesized strand, I will be able to get 50% hybrid, 50% light. Idinna separate madre, what will I get? How many blue strands are there? 2 blue strands are there. How many red strands are there? 1, 2, okay, 1, 2, amele 1, 2, 1, 2, 1, 2, okay. 8 strands I am getting. For all the 8 strands, if we form newly synthesized strands like this, okay, newly synthesized strands, what am I going to get? I will get 75% light, 75% hybrid, and I will get 25% hybrid and 75% light, correct alva? So 8 out of 8 only 6 is going to be hybrid, okay, 2 are hybrid, rest 6 are light. So here and here if I see, I am going to get, obviously understood that I am going to get 75% light, okay, 75% light, and I am going to get 25% hybrid. Ivaga next step ಗೆ ಹೋಗಬೇಕಂದ್ರೆ ಇದೇ ತರ separate ಮಾಡಿ you have to do students. ಅದು approximately 87.5% light ಇರುತ್ತೆ, okay, on 12.5% heavy ಇರುತ್ತೆ, not heavy, hybrid, sorry, hybrid, okay, hybrid irutte. Understood? Clear to all of you? Yes or no? Okay, clear? So that is your Messelson and Stahl experiment. So here remember this few things, okay. What are the things that you have to remember here? Star use madana students, star, 1 star, 2 star, 3 star, yeah. So here you have to remember that it replicates in 20 minutes, you have to remember. Then you have to remember they are not radioactive isotope, not radioactive, okay, they are just heavy and light, okay, not radioactive, you should remember. Third thing, what will you remember? That these people are doing N15 to N14. Okay, N15 to N14. So we are going to get more of N15 compared to more of N14 compared to N15. Okay, now Illon's question is there, students, important. Very similar experiment involving use of radioactive thymidine. So ill nodi, idu radioactive. This is radioactive. So remember this. Okay, when they were trying to work on, they used radioactive thymidine to detect the distribution of newly synthesized DNA in chromosomes was performed by VIDYA Faba Beans by Taylor and colleagues in 1958. The experiment proved that DNA in chromosomes also replicates semi-conservatively. So they wanted to try this in eukaryotes as well, and they noticed that yes, even in ours DNA replicates in semi-conservative manner. Okay, clear to all of you? So they will ask you what did Taylor and his colleagues work on, or they will ask you what is faba beans, which is the year that they proved that, did they use radioactive thymidine. Itara different types of questions can come. Clear? Okay. This also I hope it's clear. I have shown it with color. Now, enzyme machinery, students. How do we separate these two strands? Alva, nan easy agi helta itu parent strand na separate madbekande. But for separation of these strands, it's not an easy job. We have to separate the strands with the help of enzymes, correct? So normally nam DNA banbadi itare irutte. Okay. For my replication to happen, for DNA replication to happen, I need to make sure my strands are like this. Okay, then only I will be able to work. Now, how do I go from here to here? I will be using my enzyme machinery. Okay, what will I be using? Enzymes. Students, luckily in your NCERT, they have mentioned only 2 enzymes. So, we will stick to that. Okay, we will stick to that. Now, look here. How do we separate the strands? Let's assume our DNA like this. Okay. Now, this separation happened because of an enzyme called helicase. What is the enzyme called? Helicase. So, students, helicase enzyme you will study again in biotechnology also. Helicase enzyme banbitu use enandre, this is going to help in the unwinding. Helicase unwinding enzyme anta karitivi. Unwinding enzyme. Okay, unwinding enzyme. Andre, the strands are, it will become like this. So, the unwinding job is going to be taken care by helicase. Okay, remember first enzyme that you have studied. Now, let's take example, students. Helicase has unwound our DNA to one level. Okay, to almost one level, our strands have been separated. Now, these strands separate admere, osa strands form madbekandre, we have to bring in one enzyme. That enzyme is called as DNA dependent DNA polymerase. DNA dependent DNA polymerase. Tumba important enzyme. Okay. This enzyme is going to, ouch. Sorry, my leg hit. Okay. So, okay. This DNA dependent DNA polymerase is the one who is going to help in the formation of new template. Andre nimage xerox etkombekalla. So for that, who is going to help? This fellow is going to help to make to form the new template. Now, students, there is a problem. Problems are 2-3 problems we have. Okay. This DNA polymerase has some demand. DNA polymerase has some demand. First demand is that he will only do polymerization. He will only do polymerization of and form a 5' to 3' strand. Okay, polymerization of 5' to 3' strand. Okay, if he has to do this, then it is understood that namige 3' to 5' strand work madbekanta. But DNA dependent DNA polymerase ಇನ್ನೊಂದು ಪ್ರಾಬ್ಲಮ್ ಇದೆ. ಅದಕ್ಕೆ ಎಲ್ಲಿ ಸ್ಟಾರ್ಟ್ ಮಾಡಬೇಕಂತ ಗೊತ್ತಿಲ್ಲ. ಸೊ ಎಲ್ಲ ಸ್ಟ್ರಾಂಡ್ಸ್ ಅಲ್ಲೂ ರಿಮೆಂಬರ್ in the DNA, there will be a region which is called as your ORI, okay, which is called the origin of replication. If this is not there, then we cannot do replication at all. This is called as origin of replication, okay, origin of replication. This also is okay, but still DNA dependent DNA polymerase is like, wait, I still don't know what to do. So we will be like, okay, you know what you need a primer. Okay, students, if you can remember, you please remember. So we will add a primer. So this primer is someone, okay, this primer is someone who will lead the way for the DNA dependent DNA polymerase. Okay, now this primer is attached. So DNA will be like, okay, fine, I am happy. I will continue and form the newly formed template. So it will form the newly formed template from 5' to 3' direction as mentioned. Now problem ಏನಂದ್ರೆ students, ಈ strand form ಮಾಡೋದಕ್ಕೆ ಏನು problem ಇಲ್ಲ. Okay, ಬೇರೆ color ಅಲ್ಲಿ ತೋರಿಸ್ತೀನಿ, one second. I'll write this in little more that side, otherwise it will be too clumsy. Okay. Don't write DNA dependent RNA polymerase and come for the exam. It is DNA dependent DNA polymerase for your replication. Okay. So DNA dependent DNA polymerase, yes ma'am. So this fellow aram se what he did, he formed one new template and gave. Everyone was happy. Okay, this is formed anta. So here 3' means here it will be 5', here 5' means here it will be 3'. So this is a continuous strand, no problem in that. Now where problem will come? In this strand. Right, because if I want to do polymerization of 5' to 3', I need a 3' to 5' strand. So, ill problem start madak shuru aitu. So, ill problem enandre DNA will be like, no, I will not work like that, I will not work like that. So, primer will be like, okay, I will do one thing, reverse, I will start adding from the back side. So, I will do reverse, okay. So, DNA was like, okay, fine, he did not understand that. Okay, DNA dependent DNA polymerase did not understand that, he kept adding on. Okay, after adding on, again students remember here again, because it's going in a reverse pattern, we require more energy. At the same time, it is going to take a lot of effort because it's going in a reverse pattern. Okay, again this continued, okay, continued like this. One second, stuck aagta the board students, 1 minute. Okay, like this. So ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಕರೆಕ್ಟ್ ಆಗಿ ನೋಡಿದ್ರೆ ಇದು ಪ್ರಾಪರ್ ಆಗಿ ಕಂಟಿನ್ಯೂಸ್ ಆಗಿಲ್ಲ, ಬಿಕಾಸ್ ದೇ ಆರ್ ನಾಟ್ ಕಂಟಿನ್ಯೂಸ್, ದೇ ಆರ್ ಡಿಸ್ಕಂಟಿನ್ಯೂಸ್. ಓಕೆ, ಸೊ ದೇ ಆರ್ ಡಿಸ್ಕಂಟಿನ್ಯೂಸ್. ಓಕೆ, ಇದನ್ನ ಕಂಪ್ಲೀಟ್ ಮಾಡ್ತೀನಿ, 1 minute. Okay, like this, they are discontinuous strand. Okay, so here what will be the number of the strand? Here it is going to be 3' means this will be 5', here it will be 3'. Okay, this became a discontinuous strand. So here you label this continuous. Okay, continuous strand, and this as discontinuous strand. Discontinuous strand. Students, ಈ ಸ್ಟ್ರಾಂಡಿಗೆ ಇನ್ನೊಂದು ಹೆಸರಿದೆ. ಡಿಸ್ಕಂಟಿನ್ಯೂಸ್ ಸ್ಟ್ರಾಂಡಿಗೆ ನಮಗೆ ಒಕಾಜಾಕಿ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಅಂತಾನೂ ಕರಿಬೋದು, ಬಿಕಾಸ್ ಆಫ್ ದ ಪರ್ಸನ್ ಹೂ ಫೌಂಡ್ ಇಟ್, ಒಕಾಜಾಕಿ fragments. Now, can I leave the fragments like this? Obviously, I cannot leave any incomplete DNA. So, I will take the help of one person who is called as DNA ligase. What DNA ligase will do? He will do the gluing. Okay, he will do the gluing. See, remember, space idre al new nucleotides add madodu DNA polymerase kelsa, but adanna attach madodu, gluing madodu DNA ligase. So, the gluing enzyme is going to be called DNA ligase. Okay, remember this will be again used in your biotechnology as well. DNA ligase is going to do the gluing of the enzyme. Clear? Yes. Okay, so this is the whole story. So is it very like clumsy? If it is too clumsy, I will just draw this on little more on the top. Okay. Okay. I'll draw it here for all of you. I'll do it here, okay, helicase. Unwinding enzyme, okay, unwinding enzyme, done. So now it's a little less clumsier, and then write here about your ORI. Origin of replication, okay, very important. And students, this structure here, we call this as the replication fork, okay. What do we call this as? The replication fork. This Y-shaped structure that you are seeing is called the replication fork. Clear? Very nice. Okay, done. Okay, replication fork. So, break ಆದಮೇಲೆ ಇದನ್ನ continue ಮಾಡೋಣ, because I know ನಿಮಗೆ capacity ಇಲ್ಲ 1 hour ಆದಮೇಲೆ to listen. So, $338. Okay. Hello. Ready? Did I give enough time, students? I couldn't even drink a tea. Huh? When did I give the break? I can't remember. Okay, come on. Now let's continue. Enzyme machinery. Okay, so whatever I told, it is there in this diagram. Look at this. 5' to 3', you get 5' to 3', a strand, a newly formed strand, because your DNA does polymerization in this direction. Okay, so this strand is going to be continuous. On the other side, you will find a discontinuous strand. Okay, why it's discontinuous? Because your DNA polymerase will be like, no, I will only do polymerization from 5' to 3'. I will not do any other polymerization. So we will have to add the primer in between, so the strands will continue. Now to keep them together, we will need DNA ligase, alva? Okay, we will need DNA ligase. So that is your enzyme machinery. This diagram notebody. So idu leading strand, okay, idu lagging strand. So ill nodi students, leading strand 5' to 3' agutte. Okay, just remember this numbers, it's important. Okay, ill nodi vapas nodi yen agutte 5' to 3'. This way. Arrow marks you see, this is this way continuous, this is this way discontinuous. Okay. Now DNA unzips with the help of whom? It unzips with the help of helicase. Okay. How is the new strand formation happening? Because of your DNA polymerase. Okay. Polymerase. How is these strands going to join together? With the help of DNA ligase. Okay, DNA ligase. Correct? Yes. And remember students, there are more other enzymes also, but we do not need it for PUC level. That's why we do not teach that. Okay. But for NEET level, definitely we will be teaching. Okay. How do these people join? With the help of DNA ligase. Okay, and these fragments are also called as Okazaki fragments. Okay, Okazaki fragments. We call them Okazaki fragments after the person Okazaki. Okay, after the person Okazaki. So Okazaki fragments we call, and this strand students idu continuous, idu discontinuous. ಬೇರೆ ಹೆಸರೇನು leading strand ಮತ್ತೆ lagging strand. Okay, leading strand ಮತ್ತೆ lagging strand, they have an ORI, that is origin of replication, from where replication is going to start. Okay, we will read about it in NCERT. Come on. So in living cells such as E. coli, the process of replication requires a set of catalyst or our enzymes. Okay, or our enzymes. The main enzyme is referred to as DNA dependent DNA polymerase. Okay, so first main enzyme is DNA polymerase. Okay, clear? Yes. Okay, come on. So since it uses DNA template to catalyze the polymerization of deoxynucleotides, these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides. So students, ಇಲ್ಲಿ main ಏನು ಅವ್ರದ್ದು ಪ್ರೊಸೆಸ್ ಬಂದ್ಬಿಟ್ಟು ತುಂಬ ಅಕ್ಯುರೇಟ್ ಆಗಿ ಮಾಡಬೇಕಲ್ವಾ, ಅವ್ರ ಪ್ರೊಸೆಸ್ ತುಂಬ ಅಕ್ಯುರೇಟ್ ಆಗಿ ಮಾಡಿಲ್ಲ ಅಂದರೆ ಮ್ಯೂಟೇಷನ್ ಆಗಿಬಿಡುತ್ತೆ. ಸೊ DNA polymerase ಜಾಬ್ ಇಸ್ ಮೋಸ್ಟ್ ಇಂಪಾರ್ಟೆಂಟ್ ಜಾಬ್. ಲೈಕ್ ಹೌ ಅ co-founder CEO's ಜಾಬ್ ಇಸ್ ಇಂಪಾರ್ಟೆಂಟ್ ಇನ್ ಅ ಕಂಪನಿ, ಆ ಥರನೇ DNA polymerase ಜಾಬ್ ಬಂದ್ಬಿಟ್ಟು ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್ ಜಾಬ್. ಅವ್ರಿಗೆ ಒಂದು ಮಿಸ್ಟೇಕು ಮಾಡೋಕ್ಕಾಗಲ್ಲ, ಬಿಕಾಸ್ ಮ್ಯೂಟೇಷನ್ ಆಗುತ್ತೆ. ಸೊ ಹಿಯರ್ ವಾಟ್ ಇಸ್ ದೇ ಟ್ರೈಂಗ್ ಟು ಸೇ ದಟ್ ದಿಸ್ DNA template to catalyze polymerization of deoxynucleotides, these enzymes are highly efficient. Okay, enzymes, and they have to catalyze polymerization of large number of nucleotides in a very short time. Okay, now E. coli that has 4.6 into 10 to the power 6 base pair, it okay, completes the process of replication within 18 minutes. Okay, that means the average rate of polymerization has to be approximately 2000 base pair per second. Okay, 2000 base pair per seconds. Clear? Yes or no? Now, not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. First students, our fast ಆಗಿರಬೇಕು, ಸ್ಲೋ ಆಗಿ ಮಾಡಿದ್ರೆ ಪ್ರೊಸೆಸ್ ಕಂಪ್ಲೀಟ್ ಆಗಲ್ಲ. ಅದೇ ಟೈಮಲ್ಲಿ ಅವ್ರ ಅಕ್ಯುರೇಸಿ ರೇಟ್ ಬಂದ್ಬಿಟ್ಟು ಲೈಕ್ ಪರ್ಫೆಕ್ಟ್ ಅಕ್ಯುರೇಸಿ ಆಗಿರಬೇಕು. ಅವ್ರ ಅಕ್ಯುರೇಸಿ ಪ್ರಾಪರ್ ಆಗಿಲ್ಲ ಅಂದರೆ ಮ್ಯೂಟೇಷನ್ ಆಗೇ ಆಗುತ್ತೆ. ಓಕೆ ಓಕೆ how do I pronounce your name? Geo student. Okay, thank you. Now, any mistake during replication would result into mutation. See, every word I am telling is part of NCERT. Furthermore, energetically, replication is very expensive. Very expensive andre, ma'am, how much ma'am? 1000, 2000 ataralla. ATP, energy utilized madak tumba, like lot of energy is utilized. Deoxyribonucleoside triphosphate, okay? Adanna easy words alli ಏನು ಹೇಳ್ತೀವಿ ನಾವು DNT ಅಂತ ಹೇಳ್ತೀವಿ. What? DNT. Okay, DNT, that is deoxyribonucleotide triphosphate, serves as a dual purpose, okay, in the addition to acting as a substrate. So it acts as a substrate because enzyme substrate complex formation agbeku, so it acts as a substrate, okay. Understood? Why you are not able to understand? Are you a math student? Okay, look here. Dual purposes. In addition to the acting as substrate, they provide energy for polymerization reaction. The two terminal phosphates in deoxynucleotide triphosphate are high energy phosphates. Simple students, normally now ATP we talk, no? This ATP na nan hydrolyze madre nani ADP plus PI I am going to get, right? This is a high energy phosphate. Okay, this is a high energy phosphate. Aatarane DNT, okay, deoxynucleotide triphosphate. So this triphosphate, no, DNTP, okay, DNTP, this triphosphate also, remember, yo, why did I write capital? So sorry, I wrote capital by mistake, okay, DNTP, okay, DNTP, small letter you have to write. So this also students, triphosphate alva, so I will be able to get PI plus PI, error PI, no, high energy PI, okay, high energy PI. So it's a very energy consuming process, okay, yes, clear? Okay, English version will happen on NEET English, okay. So one English channel, one Kannada channel. Yes, Pramod, hi Pramod. Okay, now look here. So understood what is this? So DNA polymerase is the enzyme that we use. It will polymerize, polymerization will happen 5' to 3'. It will form a continuous strand. The other strand will be a lagging strand or a discontinuous strand, okay. Adamele ಏನು ಹೇಳಿದ್ದಾರೆ 2000 base pairs per second, because high fast ಆಗಿ ಮಾಡಬೇಕು, at the same time high accuracy ಇರಬೇಕು. So fast ಆಗಿ ಮಾಡಬೇಕಂದ್ರೆ 2000 base pair per second, ಮತ್ತೆ high accuracy ಮಾಡಬೇಕು, high accuracy has to happen, ಇಲ್ಲಾಂದ್ರೆ mutation ಆಗುತ್ತೆ, okay, mutation ಆಗುತ್ತೆ. Now students, energy highly expensive process, so ಅದಕ್ಕೆ energy ಬಂದ್ಬಿಟ್ಟು DNTP ಎರಡು triphosphate split ಆಗೋ ಟೈಮಲ್ಲಿ high energy yield ಆಗುತ್ತೆ, okay, that's all. Okay. Now, in addition to DNA-dependent DNA polymerase, many additional enzymes are required to complete the process. Okay, process of replication, which higher degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length, the replication occurs with a small opening. That is called as the replication fork. I taught you people this. The DNA-dependent DNA polymerase catalyze polymerization only in one direction, that is 5' to 3'. This creates some additional complication at the replication fork. Consequently, one strand becoming, okay, one strand with the polarity 3' to 5', the replication is continuous, okay, and the other one is discontinuous, okay. So simple idu madi, okay, time. You want molecular basis of inheritance? Today at 6 o'clock I have a class on NEET English, so if you want to join, you can join, okay. So illi nodi students, the discontinuous synthesized fragments are later joined by an enzyme called as DNA ligase. So DNA polymerase ಮಾಡೋ ಆ strand ಇಲ್ವಾ, ಅದು 5' to 3' direction ಅಲ್ಲಿ ಇರುತ್ತೆ. So ಆ ತರ ಒಂದು strand formation ಮಾಡಬೇಕಂದ್ರೆ we have to work from 3' to 5', right? So that is the simple understanding, okay. Yes, clear, okay. Because of the requirement of origin of replication, that a piece of DNA is, if needed to be propagated during recombinant DNA procedure, requires a vector. So, again now biotechnology will complete a goats thing, like I will revise all of that. Ivaga, don't break your head, okay. Furthermore, not every detail of replication is understood well, because in eukaryotes, the replication of DNA takes place at the S phase, okay. Nimage already go to G phase, S phase, G1 phase, alva. So they are telling at S phase it is going to happen, okay, at the S phase it is going to happen. Now the replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results in polyploidy, that is chromosome anomaly, okay. This you will study in Principles of Inheritance. Basava sir will teach you more about what is, polyploidy. You will learn the detailed nature and process in the higher classes. Clear? Yes or no? I hope this is clear to all of you. Just let me know in the chats if it is clear. Okay. So students, I will have to stop the class here. Okay, because another teacher will be taking the class here. Okay. So that's why I'll have to stop the class here today. Next class alli illinda continue madana, transcription, translation, chapter mugisana. Okay, one more class we will be able to complete the chapter. Okay, clear? Yes. So this notes and the part 1 notes you will be getting it on the Telegram channel. Now what I want all of you to do is quickly subscribe to the channel and okay, mark your attendance as well. Okay, mark your attendance as well, because I know you people are very confused. You will not mark your attendance, so mark your attendance. Clear? Yes or no? Huh? English version. Okay, so English alli if you want to watch neat content, proper neat content nodbekandre physics, chemistry, biology ge namage innond channel ide, neat English channel. Ee channel alli mainly PUC focus matre KCET focus irutte. Neat ಮಾಡ್ತೀವಿ, ಮಾಡಲ್ಲ ಅಂತ ಅಲ್ಲ, ಮಾಡ್ತೀವಿ, ಬಟ್ ನಮಗೆ so biology, me and Basava sir are neat teachers, but adik PUC teachers also, even Pramila ma'am is a neat teacher, Catherine ma'am also, so we can do a neat classes, but first main focus ಬಂದ್ಬಿಟ್ಟು PUC, because PUC ಗೆ ಚಾನಲ್ ತುಂಬ ಕಡಿಮೆ ಇದೆ, where they teach in a very simple way, so ಅದಕ್ಕೆ mainly PUC focus ಮತ್ತೆ KCET focus. neat ಬೇಕಂದ್ರೆ we will do. Okay, clear? Thank you so much students, thank you so much, see you all in next class. Okay, so bye bye, take care. I wanted to complete transcription also, but unfortunately namak studio illa, so adike navu ivaga ಇಲ್ಲಿ stop ಮಾಡಬೇಕು. Okay na? Bye bye.